DNA damage is a mechanism of disruption. So DNA damage due to environmental factors and normal metabolic processes inside the cell occurs at a rate of 1,000 to 1 million molecular lesions of per cell per day. So while this constitutes only about 0.000165% of the human genomes in approximately 6 billion bases, approximately 3 billion base pairs that can, if left unrepaired, can cause mutations in critical genes and also can cause cancer. So that is why DNA damage is called a mechanism of disruption. And what are the objectives? So first is to understand and explain what is DNA damage. Second is to know the types of DNA damage and what are their mechanisms and how they cause diseases. And lastly, to understand and explain the importance of DNA repair mechanisms. So the DNA damage. The human genome constitutes approximately 3 billion base pairs and experiences molecular lesions per day due to environmental factors and metabolic processes inside the cell. And again, if left unrepaired, this could cause mutations in the critical genes, such as tumor suppressor genes, that can hinder the cells to carry out its function and increase the possibility to develop tumors and cancer cells. And the vast majority of the DNA damage affects the primary structure if the double helix or bases are chemically modified. And what happens in the picture is an example of a normal cell that a cell attacked by free radicals, and it forms a cell that are bombarded with oxidative stress and followed by DNA alkylation or then the base loss. So these are just some examples of DNA damage mechanisms. So what is DNA damage? It is an alteration of the chemical structure of DNA, such as a break in the strand of DNA and a nucleotide missing from the backbone of DNA. And it can occur naturally or due to the influence of environmental factors. And DNA damage is an abnormal chemical structure in DNA. While mutation is a change in the sequence of base pairs. So there is a difference between DNA damage and mutation. So DNA damage focuses on chemical structure in DNA, while mutation focuses on the sequences of the base pairs or DNA. And the bases can be damaged by oxidative processes, alkylation of bases, base loss caused by hydrolysis of bases, DNA cross-linking, and DNA strand breaks, the single and double. So the first mechanism of DNA damage, the oxidative damage. Reactive oxygen species can cause significant damage in cellular stress, in which hydroxyl radicals or OH group can be produced by ultraviolet and ionizing radiations. And this results in the formation of 8-oxo-7-8 dihydroguanine or 8-oxo-G from guanine residues, and increased levels of 8-oxo-DG as association with carcinogenesis and different diseases. So what happened is that the guanine residues will oxidize, in which a double bond is added to the ring structure of the guanine, forming an 8-oxo-G, and it can cause neurodegeneration, including Alzheimer's, Parkinson's diseases, carcinogenesis, such as leukemia, melanoma, breast cancer, prostate cancer, colorectal, gastric, lung, and ovarian cancer. And another OS-induced DNA damage includes cardiovascular diseases, diabetes, Wilson's disease, Huntington's disease, acquired immunodeficiency syndrome or AIDS, inflammatory bowel disease, and chronic obstructive pulmonary disease and rheumatoid arthritis. Next is the alkylation of DNA bases. So it is produced indigenously as byproducts of cellular metabolisms. So these introduced lesions into DNA or in RNA bases are mutagenic, cytotoxic, or neutral to cell. And alkylating agents such as methyl and ethyl modify base pairs. And the most common types of DNA alkylation includes N7-methylguanine, N3-methyladenine and O6-methylguanine. So these colored molecules, the oxygen and the oxygen, nitrogen, the colored green are minor modifications only, while those that are in yellow are major modifications caused by DNA alkylation. And the same thing happened in this chain, in this monomethyl phosphotriester RS isomer, in which a methyl group is added within the trigonal planar outline outside, that forms a link between the two ring structures. And the DNA damage, the base loss. It is a location of DNA and RNA that is neither purine or pyrimidine base, either spontaneously or due to DNA damage. And they are called AP sites or apurinic or apyrimidic sites. And this causes replication, fork stalling, and are often bypassed by translation synthesis. So a type of mechanism is depurination, in which the hydrolytic removal of guanine or adenine from the number 1 carbon of deoxyribose in a strand, where DNA glycosylases act as repair. So in base loss, so there is a loss of a base in which it makes the sequence incomplete, so that causes DNA damage. So what happens in DNA in depurination? So it would be replaced by another base in order to complete the sequence again, and will be optimally function. In the DNA damage, DNA cross-linking. So this occurs when various exogenous and endogenous agents react with two nucleotides of DNA, and it forms a covalent linkage or covalent bond between the two strands, and can occur within the same strand or the intrastrand between opposite strands of this DNA, or the interstrand. And this interfere with the cellular metabolism, such as DNA replication and transcription, triggering cell death or apoptosis. So intrastrand, so occurs in the same strand from 5' to 3', and interstrand is occurs between opposite strand. So interstrand, opposite strand. So from the strand of from 3' to 5', from the strand of 5' to 3'. So that's how DNA cross-linking cause damage within the cell. Next, the DNA damage mechanism is the DNA strand breaks. So ionizing radiation that are created by radioactive decay in cosmic rays causes breaks in DNA strands. So this is called by the radioactivity or the radio decay of this kind of radiations. And interstrand crosslinks can also lead to DNA strand breaks if the damaged DNA undergoes DNA replication, and covalently stalled topoisomerases remain linked to DNA, which undergoes abortive catalysis for single strand breaks and double strand breaks, and serve as a damage sensor. And the resting state of gene expression is that there is a transcription starting site in which the nascent the polymerase 2 is in a pause where the nascent RNA cannot be replicated, and induces positive supercoiling, in which it turns the DNA going to the right direction, squeezing tightly the DNA, and that it causes damage, forming a 1+1 nucleosome. And also it is caused by stimulus-induced transcriptional activation, followed by TOP2 beta CCS mediated DSB that cause transcription-coupled DNA damage response. And DNA repair mechanisms. So we have DNA damage mechanisms. In order to repair them, we have also these mechanisms. So the single strand break, single base damage, bulk collisions, crosslinks, base mismatch, and a double strand break. So these are examples of DNA repair mechanisms. and DNA repair maintains the cell's integrity of its genetic code. And this ensures the survival of species, enabling the parental DNA to be inherited to the next generation, which preserves the health of the individual. If left untreated, it may lead to DNA damage and diseases. So the first DNA repair mechanism is base excision repair. So these are specific DNA glycosylases that catalyze hydrolysis, or the addition of water into and breaking the bond between this molecule in order to create to separate them into another molecule, in which the damaged base from affected deoxyribose in the DNA. In apurinic or AP endonuclease recognize the deoxyribose with the missing base and nicks or seals the DNA at the nucleotides. And the phosphodiesterases are enzymes that hydrolyzes remaining ester bond of sugar phosphate, in which the DNA ligase 3 seals the remaining nick in the strand. So there is a base loss here. Instead of cytosine, there will be substitution of uracil by deamination, and the uracil DNA glycosylase is the responsible for the repair. And the AP endonuclease nicks at the missing base, where phosphodiesterases removes baseless sugar, and DNA polymerase adds back C, and DNA ligase seals the nick, and the repair is complete. Next, the nucleotide excision repair. So this includes the removal of thymine dimers and recognizes the pyrimidine dimer and hydrolyzes phosphodiester bonds. And DNA helicase, polymerase, and ligase are important parts of these mechanisms, in which in helicase, this unwinds and separates the DNA fragment containing the dimerized bases from damaged DNA strand, and DNA polymerase acts 5' to 3' to fill in the gap, and the DNA ligase seals the gap in which the remaining nick in order to complete the repair. For example, there is a dimer present in the sequence between the cytosine and the thymine base, where excision nucleus cuts at arrows in helicase, which is damaged fragment, and forming a gap, 12 nucleotide gap, and the DNA polymerase fills in the missing nucleotides. And once is sealed or nicked by the DNA ligase, the repair is complete. The next mechanism is the mismatch repair. It is similar to excision repair, except the DNA defect is a base pair mismatch in the DNA, not an abnormal nucleotide, and occurs during DNA polymerase proofreading that misses an incorrect base insertion. And once recognized, a segment of the DNA in this region is removed, and repair shows that the pattern of methylation, where methyl groups remain the template of DNA during replication, but the heavily synthesized DNA is unmethylated. So when nucleotide mismatches occur in replication, mismatch repair efficiently fixes the problem about 99.9% of the time, and there will be detection that happens on the proofreading section, and during proofreading, the base that is the incorrect base will be replaced or will be resynthesized again and will be pasted with the DNA ligase, and the mismatch would be repaired. Another DNA repair mechanism is non-homologous end joining, or the NHEJ. This occurs in the double-strand breaks, where nucleotides are hydrolyzed from both strands at the break site, leaving blank ends, in which several proteins bring DNA strands together and further hydrolyze single DNA strands to create complementary strands. DNA ligase, once again, seals the edge bonded of the overlapping ends of DNA strand, rejoining the broken ends. So double-stranded DNA and an endonuclease enzyme will be that induces catalysis of the 5' to 3' damaged end of DNA and forming a nick on the site of hydrolytic damage, and the proteins will bind the deleted ends of DNA in creating the overlapping complementary ends, then and undergoes ligation to end join the deleted DNA strands. Next, DNA repair mechanism. So the homologous recombination. It occurs at double strand breaks or when DNA synthesis creates a gap during replication. The strands of a normal sister chromatid are used to repair a damaged sister chromatid. So it involves both sister chromatids. So the other pair is the one who will repair the damaged pair. So the single strand breaks are repaired through the REC-A proteins, 5 to 5 3' exonuclease REC-A nucleoprotein strand invasion, where double strand breaks are repaired with the aid of the number of enzymes in other proteins. Examples of proteins such as MRX, MRN, SA2, SGS1, XO1, DNA2, RPA, RAD51, and DMC1. And the accurate repair of homologous recombination, in which the leading strand template involves breakage in the DNA synthesis, repair the leading strand template where it is the new leading strand and the formation of the new replication fork, then continued replication, in which, as a result, there would be a repair by homologous recombination that leads to the accurate repair of damaged DNA with no deletion. And that is the end of my presentation, and thank you.